today we've got Dr. Moriah Bilenky with Purdue University to talk about small-scale livestock and soil health. And we've also got another speaker, Anna Paltseva with Purdue University, to talk about lead contamination and urban soils. So we'll jump right in, and we're going to start with the small-scale livestock. So I'm going to stop sharing my screen so we can allow Moriah to share hers.
Mm-hmm. Give me 1 second. Mm-hmm. Can everybody see all that?
Yes.
Great. Well, thanks for having me today for the urban soil updates. Again, my name is Moriah Bilenky. I started in the HLA department in January 2023 in sustainable vegetable production, and my lab focuses on basically applying agroecosystem principles to horticulture production. So trying to find and identify sustainable cropping systems that achieve farm and farmer resilience. So it's just a picture of me and some of my lab mates from this summer. So just to give a little bit of background on what are integrated crop livestock systems, and I guess there are 3 kind of main ways that integrated crop livestock systems have been discussed before. So one is at the field level, basically where crops and livestock are in the same field at some point or other, not typically overlapping, but can be, but otherwise in crop rotation, which with each other. But then we can also talk about crop livestock systems at the farm level, where the system is not necessarily, where crops and livestock aren't necessarily sharing the same field, but where crops and livestock are produced on the same farm, and manure from the livestock system is being used in the cropping system, and forages or plant material from the cropping system is being used for animal production. So still those 2 systems are benefiting each other. But we can also talk about it at the region level, where farms in the same region, one producing livestock and one producing vegetable production, are exchanging these nutrients across the farm. So again, manure from a livestock farm going to a crop farm, and that same farm having plant material for forage or feed going to that farm. So primarily in the Sustainable Horticulture Lab, we mostly are focusing on the crop livestock integration at the field level. I see some things. Okay, in the chat. Okay, so crop livestock systems and soil health, there have been many cited benefits on use of crop livestock integration, but as far as soil health is concerned, research has noted improvements in aggregate stability, from having animals grazing. Oftentimes this research is looking at perennial pastures as well. So it's usually the combination of grazing on perennial pasture that's improving the aggregate stability, increased total carbon, in some cases soil organic matter, but usually only in long-term studies, increases in total inorganic and organic nitrogen, typically inorganic and directly after animals have been removed from the plot, which makes sense. You know, they're depositing manure on the field and things like that. In some cases, increased soil respiration and microbial diversity have been seen as well, but this can be much more variable. Okay, so we've been doing some research on crop livestock integrated systems for horticulture production systems, and some of this work started when I was at Iowa State in grad school. And so that one project was integrating pastured meat chickens into organic crop rotations. And so the idea for this was that, you know, in organic, where you're limited to allowable or National Organic Program allowable sources for inputs, including fertilizers. So, you know, organic commercial fertilizers or manure and composts are typical sources that are allowed. And so could we create more of a closed loop system by including pasturing meat chickens into the crop rotation, as well as diversify the farm income stream by growing broilers on the same farm. And so what we did was, this was an annual crop rotation that went for 3 years. And so these were on small plots. So the idea was this would be applicable to small farms. So each plot was 15 by 25 feet, roughly. And we grew spring vegetables. So we had 3 different rotations each year. But typically we had a spring vegetable, which was followed by cover crops, and then poultry grazing on cover crops, or vegetables with no chickens after, or chickens directly after the vegetables, so to facilitate residue cleanup. And then we repeated the rotation for 3 years. So chickens always followed the vegetable production in the basically late summer or fall, and vegetable production was in the spring. And we used these movable coops, and you'll see a few pictures, and you can see one of the pictures there, but they were 5 by 4, and we stocked them with about 10 birds each. So they had no, we had 10 birds each, so there was about 2 square feet of floor space per bird in these. And each year we used some we used either Red Ranger or Imperial, which are both slow growing breeds, ideal for more of a pastured system. They have stronger legs, so better bone strength, and are more active typically in the field. And then we measured soil health at different time points. So primarily we looked at, again, we were trying to see if fertilizer inputs could be reduced. And so primarily we looked at inorganic nitrogen, but we also looked at organic matter, microbial biomass carbon, and things like that. And so we sampled at 4 time points to get sort of baseline post chicken data. And so this is from our 3 years of data. We didn't really see much difference between the treatments in the first year. In the second year, we saw an increase in inorganic nitrogen, as well as in the compared to the plots where no chickens were. So this VCC treatment is vegetable cover crop without poultry. And VCCP is the vegetable cover crop with poultry grazing the cover crop. And VPCC is where chickens were right after vegetable production, facilitating cleanup with cover crops planted after. And in some cases, we saw higher inorganic N after the removal of the chickens. And part of this probably that we didn't see this difference in the first year in 2017 may have been the chickens grazed for a little bit of a shorter amount of time than in the other 2 years. And then this is so these were the different time points. So I should just go back and show you. So this was midsummer. So this would have been after we removed chickens from the vegetable poultry cover crop system. So which makes sense that there was much higher inorganic N at that time point, whereas the end of season sampling time point, where we see higher nitrogen here, is because chickens have just been removed from this vegetable cover crop poultry system. And then we also, again, we looked at microbial biomass carbon, and it was variable. So like in the first year, we didn't see any differences among the treatments. So again, summer was when chickens had just been removed after grazing on vegetable crops, whereas autumn is when chickens from the poultry cover crop grazing treatment had occurred. And so we saw some increases over time with microbial biomass. And then in year 3, we finally saw some differences in the different treatments themselves. And this also might just be that, the animals themselves didn't provide that much of a difference over the application of the cover crops in those first years. So currently we're working on another project here at Purdue. This is an organic center FFAR funded project. And we're looking at investigating the reintegration of organic food, food crops, and animal production. And it has a similar theme. Again, we're looking to see how we can increase or reduce the use of off-farm inputs in organic cropping systems, as well as measure soil health and above and below ground biodiversity. And we're looking at 2 different systems in this project. One is a chicken integrated system, very much like the one I presented, and a sheep integrated system. And so we have 2 locations of the sheep system, one at Meg's Horticulture Research Farm and one at the Southern Indiana Purdue Ag Center. And the goal of this was to have to look at how legacy of the farm might affect the parameters we are measuring. So Southern Indiana is a history of perennial grazing and perennial crops and no annual vegetable production, while Meg's Horticulture Research Farm is in an annual crop production for quite some time. And so these are what the two different plots looked like. So this is at Meg's. And this is 145 by 405 feet. And the area we did the study on here in Southern Indiana was about 100 by 140 roughly. And this study is currently ongoing. This is the first year of the study, and we started it in March of this year. And so in the spring at Meg's, we established a cover crop of oats and peas for grazing. And at Southern Indiana, we just grazed whatever cover was already there, which was composed of perennial ryegrass and alfalfa and a few other perennial species. And so this is what we did. So at Meg's, this was a randomized block design with 4 replications. And so basically we had 4 grazing paddocks and 4 no grazing paddocks. And we stocked the paddocks with 8 sheep per paddock. So, and each one was about 100 by 90 feet. So we had 8 sheep on a 100 by 90 foot paddock replicated 4 times. And then Southern Indiana was very similar, except since we had smaller space, we did just three replications, and each plot had 4 sheep in each one. And so this is what the plots looked like during grazing time. So we grew the cover crop, and we seeded the cover crop in mid-March, and then put sheep out on the plots at Meg's in May. And in Southern Indiana, we had the sheep out in mid to late April. And this is what the plots looked like when the sheep were basically first put on, and this is what it looked like just prior to removing the sheep. And just another picture of them grazing out at Meg's. And actually in this picture where it's all in the front here, this is all thistle, tall thistle plants that they removed all the leaves from, which was another interesting part. And so then after the sheep were removed, we turned the plots over and we had 2 treatments, no-till and conventional tillage. So basically we had plots that had been grazed that were then tilled or no-tilled, and plots that had not been grazed that were tilled or no-tilled. And the no-till plots that were not grazed, we used tarps to terminate that cover crop. And so this is just showing what those plots looked like after removing the tarps. And this was in Southern Indiana, where there was all that perennial, well-established perennial forage. And so we then planted tomatoes, and so we're still currently harvesting all those tomatoes in these plots. We put 2 rows of Roma tomatoes in each plot, we're using organic methods since it's an certified organic land, and we've just been harvesting them and collecting yield and quality data from the tomato plants. So all through these different time points, we've been taking soil samples. We took a baseline sample at the start of the study, and then we've been taking, we took one sample post-grazing in all plots, and then we took another soil sample at the time of harvest. And so the chicken system, it's very similar to the sheep system, except we're using chickens. We're using, again, pastured meat birds like in the previous study. But this project only occurs at the Meg's Horticulture Research Farm. And again, we're using a little bit of a larger chicken coop than last time. So but the similar stocking density. So anywhere from 10 to 12 birds in a coop that's 10 by 5 by 10, sorry. Yeah, 10 by 20. Sorry, I don't have the measurements in front of me, but it's a bit larger than the last one. Okay. And so this is what they look like here. And so we have the chickens out grazing and or foraging from about 4 to 5 weeks of age. And we're moving these chicken coops every day and measuring feed consumed and calculating feed conversion ratio for these birds. And we did have 2 additional treatments within this system. A heavily mowed and a not mowed treatment. The heavily mowed treatment, the idea for these 2 treatments was trying to understand if access to forage quantity provided any changes in feed conversion ratio. And so that was our main question for having those 2 different treatments. Otherwise, it was the same. We followed it with conventional till or no-till and tomato production. And then this is just the Red Ranger birds in the coop. Again, this is oats and peas and weeds that they're foraging on. And we provided them with a full ration of organic feed. And then this is just a video showing how the coops are moved. Sorry, it's 5 by 10. It's the size of the coop. I don't know, I couldn't think of it before. And you can see this is just electric netting that we've used around the field just to ensure predators don't come in. And then this is what the field, this is the chicken system field after it was planted to tomatoes. And this is just some of the harvest that we had in southern Indiana. We had record harvests there. We're still going through all the yield data, but over 1,000 pounds of tomatoes have been harvested from those plots there. Okay, so some of the preliminary soil health data from the sheep system from 1 year, from this 1 year, so I can move this around. So what we have here is the MEGS system and the Southern Indiana system. And so far, what we have seen is that within the treatments, and these are post-grazing. So these are the soil samples right after the sheep were removed and the plots were about to be flipped over into tomato production. And what we saw here is that there really weren't treatment differences within the sites, but there were differences in the soil health between the two sites. And this is, as I mentioned before, some of the benefits of crop livestock integration have to do with animals grazing on pasture and having that perennial forage there. And so I think primarily this is just because of the greater soil health of the perennial multi-species forage system that was already in place there. The soil health at the MEG site was just initially much lower to begin with. Specifically, organic matter aggregate stability was almost twice as high or more at Southern Indiana than at MEGS. For the chicken system, again, we saw something very similar, that there really weren't differences between the treatments. And again, that this may just be that the cover crop itself is providing is making the systems consistent across, and having the chickens there is not necessarily improving upon that, at least in 1 year. And we're also working with a couple growers on this project. So each of these growers has a side-by-side comparison on their farm. So a strip where they graze chickens in the fall or winter, and a strip where they don't have chickens at all. And we're taking post grazing soil samples there and looking at soil health with and without chickens on their farms. So far, what we've found is, again, not much difference between the two, with chickens and without chickens. However, their soil health test results were much better than the MEGs. The MEG site, which is interesting because these sites also have history of annual crop production like MEGs, but it's likely due to the high additions of compost and other types of organic matter that these farms are using, where at MEGs, the history of that farm has been, at least the site we're on, weedy fallow for the last year or two, and with no additions of other types of organic matter. And so we're also still looking for additional collaborators for this project. So if any interest in having a side-by-side paired strip on your farm, and us coming out and measuring soil health with and without chickens, as well as helping you work on a budget or enterprise budget for these two systems, feel free to fill out the survey, and we'll be in touch. Other research that we're working on, stuff that's in the works, we really want to understand what growers think of crop livestock integration and what they think might be the benefits and challenges. And so we're hoping to create a survey to ask farmers some of these questions, and help us direct our future research, as well as with talking with other farmers. It sounds like knowledge sharing is one issue with crop livestock integration, is that there's not that many folks doing it, or if there are, they're in the different places and they're not able to connect. And so trying to connect the growers who want to do crop livestock integration, and share what they've been doing and what has worked and what's a challenge, or what they hope to do, is something else we would like to do. And with that, I guess I can take the questions that are in the chat. Let's see. Okay, so first question, in the chicken veggie cover crop rotation sequence, can you share a little bit more about the manure management to reduce pathogens? What cover crop mix did you use? So, so for the study at Iowa State, we went by, and actually here at Purdue as well, we followed the organic guidelines of waiting 90 days from the time of application of manure to the harvest of that crop, for crops that don't come in contact with the soil, or unlikely to come in contact with the soil. So we applied that to the livestock integration, saying, this is the manure application, and when we removed the animals, that's when day one of that 90-day period started. And in that system, we were using cereal rye or oats in the different rotations. And then in this system here at Purdue, we're using oats and field peas as a cover crop. And again, using that same 90, 120-day rule. So the question was about difference in size of animals between treatments. So in the study at Iowa State, there wasn't any difference in the size of the birds, but in some years there was a difference in feed conversion ratio when birds were on pasture in the summer compared to in the winter. So basically in the warmer months, feed conversion ratio was not as good. I think probably because of the heat stress. Here we haven't looked at that. For this study, for this year, we haven't looked at the data yet to compare the heavily mowed and not mowed systems for the chickens. No, the data is not normalized for the ungrazed field, but all the fields had the same cover crops. And again, yeah, this is just the data I showed from this year's project is just preliminary. analysis. So yeah, there's much more to look at this winter for sure. Oh yeah, and good question. For as far as growers, how long they've been doing crop livestock integration, this was their first time rotating on the vegetable fields. So my thought is really the higher performance is due to the additions of compost that that they had been doing that had not been going on at MIGS. And yeah, I can share that link. And let's see. The data is not public yet. These are just some of the raw data. The study is still ongoing this year. Yeah. And then we have two more years of this project. Yeah.
Can I ask a question?
Yeah.
If we will put a chicken to urban spaces, and in some cities they do, I don't know if there is such a thing already in Indianapolis or any other cities that you know. And then what happens to chickens when they die? I assume you cannot just, consume them. Not when they're dead, but like during their life cycle from urban spaces. But like, what is a typical protocol of handling chicken in urban spaces?
That's a good question. And so, for this study, we were growing pastured meat chickens. So they had an end time and then we were processing them. And then after that meat could be sold. For example, we processed the chickens in this study through the meat lab here on campus. As far as if the question is about like mortality and just like if for some reason a chicken dies, what do you do with it? In those cases, like, you can put the chicken in the compost pile. For the research, if a chicken dies unexpectedly, then we need to let lab animal resources know and figure out what caused that chicken to die. But I'm not sure specifics for like how you would handle that in the urban areas. That's a good question.
Yeah, it's really interesting. Thank you.
Mm-hmm.
Yeah, Ellie answered some question.
Oh.
Thank you.
Okay, thank you so much. That was an excellent presentation. Very informative for us. So now I'll just turn it over to Anna, if you want to share your screen, and I'll let you go ahead.
Yes, just. Oh. Okay. Here it is. You're able to see it. Good.
Yes.
Okay, good. Well, good morning, everyone. Thank you so much for inviting me to join your meeting. It's really great to be in Indianapolis, learning what you're doing in Indiana in general. I'm a very new clinical assistant professor at both departments of agronomy and horticulture and landscape architecture in the College of Agriculture. And I'm learning my way about. So thank you for sharing the insights into your program. letting me to join in, and I hope we can meet at least some of you next month at the conference. Oh wait, not this month, it's next week, the conference that you organize on October 16th. So in today's presentation, we'll move to urban spaces, and I will share with you what I've done in New York City, where I did my previous research, and what I did recently in Louisiana, another Lafayette, Louisiana, at the University of Louisiana at Lafayette, and what I would like to do at Purdue. And I welcome your ideas, some contact information, anything you think I find interesting, I would really appreciate it. So when I moved to New York City many years ago, I was astonished by gardens in the middle of the city. I wondered, why is it people have to grow their own vegetables in a city full of resources? I'm originally from Russia, and my parents, or all like my family, grow their own vegetables because it's part of culture and part of really a survival strategy. You have to produce your own food. But in cities, I didn't realize that, like, why people need to do this. They have so many stores. But that's later on, when I started studying at Brooklyn College and involved in the soil lab, I've learned there are so many different benefits, and it goes beyond nutritious food for locals. It's also family bonding, neighborhood bonding. It's a grocery reduction bill, of course. There's mental health issues. And what's most interesting for me is minimizing criminal activities. There were some studies done when they built community gardens in very active areas of the cities, and criminal activities were dropped because people had something to do, as well as they would less likely to hurt someone they know or work with in the garden. So really interesting observations about creating more of these functional green spaces. And of course, it allows us for deeper connection to agriculture and nature in general. But all these beautiful benefits have potential to be contaminated, especially in the urban environments. So some metal, I focus specifically on metals. There are multiple pollutants in the soil, but here's the urban and suburban contaminant sources that we commonly find is arsenic, cadmium, chromium, copper, mercury, nickel, lead, thallium, zinc, and many others, but they come from such sources as insecticides, pesticides. It could be treated wood, can be fertilizers, sludges, runoffs from roads, emissions, of course, different leaded, lead sources like lead paint, gasoline emissions that accumulated during 20th centuries and still present today. So what we study is really a legacy contamination. And this lead, what we have is in the soil is mostly come from lead paint or leaded gasoline accumulation. There are some other sources like industry, of course, but we have really minimum of factories these days that would emit lead into the atmosphere. So it's really all from what we accumulated during previous decades. So in this presentation, I'll specifically focus on lead, that is most prevalent metal in soils that really can hold a danger for people, especially children. And it because that lead is neurotoxin, it affects cognitive development of children. It's quite immobile in soil, and it stays in mostly in the top layer of the soil where there was original accumulation of this metal. And because it doesn't really move like arsenic, it may stay in the soil profile for a very long time. It's especially important for children because children are more susceptible. Their bodies are still growing, and they don't have as large biomass as an adult. So the concentration of lead that gets into the soil or from the soil to their body has a larger impact compared if it was an adult that exposed to lead. So how these particles of lead can get from soil to human body? There are different pathways. One is through vegetable uptake, and we consume plants that were contaminated. Another way is through leaching to groundwater, and then we drink it. And then it's most important is actually direct exposure through ingestion and house dose. This is number one most dangerous pathway for metals to get into humans, followed by inhalation and maybe some dermal contact that could be very acute. It's more for people who work in some professional fields who get access to maybe some fertilizers or pesticides, or work in the fields where or labs where you can get contact like that. So most of soil gets into children or adults through inhalation or ingestion predominantly. Here's a map of New York City. Perhaps many of you have been there before, and this is the city, urban gardens in the city. Most of them you can see located in north part of Brooklyn, this area in circle, with a lot of Superfund sites or former factories, navy yards, smelters, with infamous Guanas Canal, the most contaminated one in the country. And we see the distribution of samples also centered closer to the historical part of New York, this northern part of Brooklyn. And the concentrations of lead drops as we go further from the historical center to more suburban areas. In this example, we have about 2,000 samples that were collected throughout many years at the Brooklyn College. This lab that I used to work with is run by Joshua Chen. So he has this lab for maybe like 15 years, and they still keep adding samples to the map. So we might see a little bit of shift in sample collection, but mostly people were concerned where they live, and they probably were aware of this, of the historical use in their neighborhoods. That's why we received so many samples from that particular area compared to other areas of New York. If you look something closer to home, samples in Chicago, this is the research done by Watson a couple of years ago. We can also see different distribution of lead throughout the city, some very contaminated hotspots. What is really interesting to know here is that earlier this year, EPA changed their guideline from 400 milligram per kilogram to 200 milligram per kilogram for lead found in bare soil of children's playgrounds. So this means that if less than 200 is like light brown color, most of Chicago would exceed this threshold or this guideline. We have an ongoing project in the parks of Chicago. This was funded by USDA and RCS. My student in Louisiana, we're still working with her, we actually went last year to Chicago, collected samples in multiple parks, 5 parks, to estimate the potential for urban agriculture. We specifically look for some sites that could be converted into gardens. They would be flat, with open light and surrounded trees. So it should be open space for gardening. And we find a heterogeneous soil that multiple areas, you can see here in this park, Douglas Park, that concentration of metal really varies. And it's super important for us to test before we do any gardening in order to avoid potential exposure through working with the soil, but also through uptake to vegetables, which is still an important pathway for contamination. By eating 1 time or 2 times a contaminated carrot is not going to kill you, but if someone is exposed to it for years, it may become chronic. Something else I found, you probably know Dr. Philip Healy from Indianapolis. He has done a lot of work in Indianapolis itself, and this is the maps that I found on his research site. Yards in Indianapolis, you can see darker samples, darker hotspots with high concentration of lead. again, in closer downtown area. Drip line even have a larger number of red dots. So the drip line typically has high concentrations of lead around the perimeter of the house, of the foundation, because of the lead paint or maybe something in roofs or pipes. So you will get high lead, which we also see the same in Lafayette, Louisiana. So he has done a lot of work in Indianapolis, but we also see the concentration of metals in historical sites in the downtown areas, and then it spreads closer to suburbs, which is a very typical pattern for many cities. One of the case studies we had in New York 10 years ago now, actually, this article came out that there were some gardens that kill people, or they give cancer to people. Our research group was curious to go and remeasure the entire garden, Sterling Community Garden in Brooklyn, and we found it was not the case. It was in power of media. They misinterpret data that was collected by Cornell University research group and exaggerated what was found. So what we found is that we simply mapped all the plots in this garden, and we found only one area with peach trees next to the road was contaminated. But luckily, they didn't grow vegetables there because there were peach trees that shadowed the area. But the rest of the garden was didn't have arsenic, and there were some levels of lead present, but they will still be able to grow vegetables. Yeah, they mostly grow vegetables for consumption. Because there's certain levels are still allowed for growing even root vegetables. So what we also did is collecting samples and determining there was a high concentration of lead and arsenic in the Duke Farm in New Jersey. And we established that it's not only urban problem, but also suburban problem. And here it was an issue with using lead arsenate pesticides back in the day, early 20th century. They stopped using it by the middle of 20th century, but legacy contamination is still there. So we did this 2016. I think this were experimental trials with my students, published this paper a few years after. We analyzed samples, determined what was the initial concentrations of lead and arsenic, phosphorus concentration, multiple other parameters, and then we added different amendments, different phosphorus amendments and different composts. And we grew multiple vegetables, leafy greens, and actually we had lettuce, spinach, carrots, radish, eggplants, and tomatoes. And one day when we came to collect the samples, our veggie samples, spinach was gone. We don't know what happened to it. Seems like someone ate all the rows of spinach. But we still got good results from the rest of the plots and give us a good idea what is the uptake mechanism and contamination pathways for leafy greens, root vegetables, and fruit vegetables. We also found a different relationship between amendments and plant type. So what we found is that plant type, what we grew, was really important. What we put into the soil to change the quality of the soil, or soil chemistry, to reduce availability of metals for uptake by our plants. Plants had different response regarding of the amendment type. What we found that root vegetables and herbs and leafy greens would have higher concentration of metals compared to leafy greens and compared to fruit vegetables. We did this analysis not only Duke Farm, but also in Sterling Community Gardens. We grew a large variety of vegetables in the plots. And this finding is also confirmed by other universities, especially Cornell University. They are one of the leading groups on urban soil health, as well as vegetable uptake of contaminants. And specifically what happens is that fruit vegetables prevent contaminants going into their own in the body of the fruit body because of strong physiological barriers inside of the plant itself. So anatomically, plant prevents from it going into tomatoes or eggplants. Plus, such plants like peppers, eggplants, tomatoes, they're tall plants that don't get enough splashes from surrounding areas, strong physiological barriers inside of plants, and their fruits typically very smooth, so you can easily wash off soil particles or dust, which is not the case for leafy greens or herbs. They have a very large surface area, very often hairy or like rough surface that allows for dust and in particles from surrounding areas to adhere on the surface, and it's very hard to wash it off. So when you wash vegetables, you reduce contamination on them, but you can completely wash it off. And then root vegetables. We found that arsenic would be found mostly in radishes, while lead was predominantly in carrots. So there's different behavior of metal uptake in different plants. And for carrots is also a very unique situation because it's not only that they accumulated in the peel, they also accumulated in the core. So it's really tricky to eat contaminated carrot because you can't really peel not only the surface, like the skin of the carrot, but also cut out the core inside. And there were some studies done by USD and Cornell when they look at different parts of carrots and really determined that it's a different distribution of lead in carrots. But doesn't mean you should stop eating carrots. Again, it's about diversity in food and your diet. Consuming different vegetables, dairy products will really help to mitigate the potential contaminant intake. So really important for us to consume more calcium that will prevent the uptake of lead by our bones. This previous experience working in New York City in the Brooklyn College while I was doing my doctorate, I moved 24 years ago during COVID to Lafayette, Louisiana, where I opened up a lab, Delta Urban Soils Laboratory. And this is the place where students conducted their research with me, but also it was a teaching lab. We had lots of fun, but most fun part was working with different communities. So it was not just research and teaching lab, it was really hub for testing soil for communities in Louisiana and beyond. And our lab focused on 4 pillars: sustainability, environmental stewardship, community engagement, and science communication. I strive to share results that we get in our research groups with not only other scientists, but also with communities and artists and gardeners and practitioners. So I like to say that my science is people science, and we really need to make it accessible in understandable way, because very often scientists can write in jargons and very complicated language, and for gardeners it can be very hard to sort through all those technical terms and find mitigation strategies. This is something you've heard a lot in New York from people who attended our workshops or came to events where we would test soil right in the spots outside as part of like soil shop events with our X-ray fluorescent analyzer. It's like a portable gun that we can just use a trigger, click on it, and it will analyze sample within a minute to give us results for multiple heavy metals. So a lot of people told us that they get lost in scientific literature. This is why I really try hard to digest scientific findings, at least for my research, to larger groups of people, and publish not only in scientific journals. So here's a case study from Lafayette, Louisiana, where we received the funding from Louisiana Board of Regents. My student who graduated this summer, Holly Hefner, she conducted this study around the city. Lafayette has about 120,000 people living in it. So, and it's very like it's very vast city. It's mostly one-story homes, except downtown with like few buildings that have like 5 floors, maybe 5 stories. What we did here is that within a couple of years, 2-3 years, we collected samples. Students would bring samples to my classes. So it would be like undergraduate research experience in the class. They would collect their samples from home. We would analyze them. But then in addition to it, Holly would go to certain areas, like different zip codes, and collect samples from public spaces. And we also worked with the Lafayette Consolidated Government, who gave us access to some private properties and parks to collect more samples. And we participated in multiple events throughout the city at the different artworks or plant sales or some festivals where we could invite people to bring samples to our lab, or allow Holly, give them her address, really allow her to collect samples from their backyards and front yards. So here's this diagrams of like schematic diagrams of how we collected the samples. If it was the house, you can see in the top panel, we would collect samples around the drip line, and it showed the highest concentrations. Would have a front yard, front lawn if there was one, in a backyard. If there was separate gardens, the vegetable garden, the students would collect samples there as well. And if we did the park, when we did the grid line, the parks were collected mostly by students. There's some couple large parks, so we just went there. They we showed them a map where they should go and collect representative samples, and we did it in a number of classes. So there was a lot of involvement, multiple students, so lots of, I think it's pretty much a community project at this point. So here's the students mixing in the samples, because they would collect samples from 5 spots to make it one representative sample. They would, of course, clean the supplies in between samples, and then we would put it in Ziploc bags, take it to the lab, and dry samples, grind them to less than 2 millimeter size, and then analyze with X-ray fluorescent analyzer, and then map the city. I'll show you lead map, but we are currently working on other metals to see the distribution of arsenic, zinc, copper, and multiple other elements. So here's the lead concentration map. You can see the hot spots here in the brown, dark brown areas. Again, this is the historical part of Louisiana, of Lafayette. Historical part, it's inner city, the downtown area, where used to be lots of industry. This is Sanborn map. If we close look, we will find it's mostly all the factories or laundry, or even parts of interstate and road, and also gas stations. So, and homes that were built before 1978 with old foundation, you'll find high concentrations of them. So what we found was interesting but not surprising, that residential samples would have much higher concentrations of lead compared to public spaces, which was also found in other cities. My colleagues at Columbia University did in New York and Brooklyn very detailed study on residential public areas in Greenpoint, and they also found the backyards of private homes had high concentrations compared to public spaces, and most likely because in public spaces there are different agencies and profit organizations, government, who put compost, they grow lawn, there's constantly influx of new materials into public areas, while residential samples or residential spaces, they're not touched by people. Very often it's renters, so they don't, especially like some students told us they had a lot of garbage in their backyard, but they would rent the home, so they're not going to do anything. But those are the backyards with high concentrations of multiple metals. And we'll look at correlation between soil lead and housing before 1970s. It was hard to find a specific age of buildings, so we had to go with like some medians that we found per decades, and what we see that there is a correlation between housing that were built in before a lead ban in 1978 would have much high concentrations of lead. We also find that there's a high concentration of lead near a house, so the drip line around the foundation and street side, so it's next to vehicles, obviously, and the concentration would be much lower at open space like front lawn. We also found that soils that would be original, so meaning unamended, would have high concentration of lead, and amended soil with compost or mulch or raised bed soil would have much lower concentration of lead, suggesting us or proving actually that amendments and raised beds work for mitigation of lead. Here's the overall scope of our project. We did a lot of outreach to communities through different events. We even reached out, or actually newspaper reached out to us because one of the event meetings we attended in the neighborhoods. Then they wrote the piece, then they made a video on a local channel about our project. That would allow for more samples to come into the lab. So we also published one paper. We also made a digest for people to understand what's in the paper without like reading the whole science paper, but know what's in the soil. We did the workshops and the webinars for those who are interested in project results. Here's a detailed report, how people would receive it. Holly shared a lot of information on the Facebook groups, but also we send individual reports to people who shared their home addresses and their email addresses if they wanted to receive results back. So we'll tell them what's in their soil, in the samples, and what can be done to improve it. So what can we do to make our soil better? In New York, there was a really great program that could be replicated in other cities and states, as New York City Clean Soil Bank, when they use a glacier sediment from excavation of new foundations for buildings in New York City. So they collect sample, collect the sediment, put them in some storage facilities where a community community-based organizations can come and pick up trucks of this clean sediment and distribute into their gardens and properties. It's sediment, so it means it's not soil. It doesn't have organic matter in it to grow vegetables, for example. It's in a garden, so they had to add something to make it more fertile. And typically, they would mix it in New York City compost or any other compost, but New York City Compost Project provides a free compost for organizations and individuals as well. So those are free resources that can be easily accessed in New York City, maybe something similar in other cities as well. There are some emerging amendments. It's basically a lot of like multiple organic matter sources can be converted into compost. You can see here some examples of what we can add into the soil to improve its health, but also mitigate contamination. So solution to pollution is dilution. So if anything we add in terms of organic matter to the soil, it helps to bind lead, making it less toxic, it less bioavailable, available for organism uptake, be it in humans or plants. And it's really important for us to add this organic matter to the soil because it has multiple multiple functions, services, but one of the major one that I'm interested in is remediation. And organic matter not only binds lead in soil, but it also creates dilution effect in our produce. The vegetables that grow in soil that is composted would typically provide larger yield, larger biomass of, let's say, tomato or cucumber or carrot, and the concentration of the metal would be diluted because there's a more water in this vegetable. So larger biomass leads to lower concentration, and that causes the second dilution effect from the compost or organic matter in general. So here's the guideline strategy, simple strategies that we share with people. You can use your phone and scan this QR code for open access article to read more about legacy-related urban gardens and how we communicate risk and limitations. In general, again, we need to add organic matter. It can be not just compost or mulch, but biochar, biosolids. It has multiple effects, functions for soil quality, like reduction of resuspension, improvement of soil health, sequestration of carbon, formation of aggregates. We can also add lime to acidic soil to make soil more neutral, at neutral pH, lead, particular lead, other metals as well, the least available for uptake. Conservation tillage help us to reduce erosion. It lowers organic matter decomposition rates, so it can remediation can last longer, and it also increases aggregate stability and reduces resuspension of fine particles into the air. We should not build gardens closer than 50 meters to the traffic, and use drip irrigation to reduce emissions. We did multiple events around in New York City. I already mentioned we did some community organization events. We did multiple events in Lafayette. This is my lab. We did it outside this event in one of the parks for Earth Day, where we would involve kids to play with dirt and lead paint. So it was really fun activities, but it was for us also to collect samples, to tell people that what we are doing, as well as educating communities. I'm excited to share with you that recently I published Urban Soil Guide that can help people to test samples on their own, not for heavy metals because you still need to send it to the lab, but for other activities like pH and salts and texture in quite digestible ways. That 20 multiple activities can be done in a classroom or in the garden by practitioners. And I try to make this as accessible as we can, because people need to be able to test what's in their properties at low cost without sending to the lab. So you can get the book if you're interested as ebook or hard copy through Springer or Amazon, Barnes & Noble, and multiple other stores. So at Purdue, I would love to study more contaminants, not just heavy metals, but move to microplastic and PFAS. I would like to evaluate multiple soil amendments and the effect on remediation. I would like to explore how climate change can impact soil management and crop health in urban agriculture. Study soil contamination in regard to food insecurity and public health, specifically in marginalized communities. Of course, collaborate with multiple departments, experts in urban planning, public health, engineering, social scientists, and climate scientists, and provide these services, potentially building a lab, soil testing lab at Purdue, where students can test the samples so they get training, but this service would be available for people in Indiana and beyond to test their own home gardens or wherever they want to test urban farms. So thank you for your attention. Here's my contact information. If you would like to stay in touch or ask me questions, I'll be happy to get involved with the projects, come out to speak to more events, or just talk about soils over a cup of coffee. Nope.
Thank you.
I'm sure here it is. Okay, I see there's microbial amendment to either detoxify or fix the heavy metals. I don't work particularly with microbes, but it's a great great avenue to explore. And some of my colleagues, I co-author some papers with them who are looking into microbes. So I think it has a really big potential, and also fungi. There's some studies that we're doing in Buffalo now with the with mycelium to remediate lead. So this is results will be published soon. can crops be pulled for heavy metals from soil and significant good carrots or similar taproot could be grown in short and then stick? Okay, this is a great question about vegetables. So unfortunately, there is no hyperaccumulator plant that can uptake contaminants in high quantity to remediate soil. There are some plants, not crops, but some other plants that can phytostabilize lead, meaning it can grow in their tissues. It could be trees too, like willow trees, but nothing like you cannot grow vegetables, tap roots to clean it up. They do not uptake enough, or we would need to spend hundreds of years to try to do it. It's just enough to be not healthy for our consumption, but not enough to clean up the soil. Thank you so much, guys. I really appreciate your comments, and nice to see some of familiar faces, names.
Thank you. We appreciate all of that info. That was fantastic. Well, I guess if there's no more questions, then we'll move on to our our regional updates from our specialists. Let me pull up my screen again. Okay. Okay, we'll hop into the Northwest.
So we don't have Marianne with us anymore, unfortunately. After 3.5 years, she has decided to take a position with NRCS as a soil conservationist in her home county in Warren, and really looks forward to being able to learn more conservation practices on other land uses. It was a really hard decision for her, and she will be sorely missed. And thank you, Marianne, for all of your work thus far. If you wish to reach out to her, she is still at her NACDnet email, so you can contact her if you're in Warren County and want to collaborate. We're also actively hiring for Marianne's position, so I will drop a link in the chat for the job description for the urban soil health position. If you have anyone that you think would be a great fit and really enjoys working with diversified farmers, and also with a diverse and very active job role with a lot of different things happening every day, then please reach out to us and share the opportunity. Next. And I wanted to give a plug for a new booklet that was finalized just as Marianne was leaving. It was her last project to help support, and it is Top 10 Small Farm Conservation Practices, and going into a little bit more depth on practices that are both best practices ecologically that we see on a lot of small farms, urban farms, as well as ones that align with NRCS equip funding opportunities as well. So an opportunity for us to do more outreach and help support technical assistance. So that we'll have those available for Get the Dirt, and then on our website soon. That is all.
Hey, good morning, everyone. I'm Terry Thyssen. I'm with the Northeast region of the state. We had an opportunity last week to get out to Full Hand Farm and do a farm tour with Genesis and Eli out there. And it was a lot of fun seeing what they're doing, how they're doing it. And we had a good group of people as part of the advisory committee to be able to do that with us. I really enjoyed seeing their farm in the growing season, and the beautiful picture of the radicchio is in the bottom left-hand corner. It was just stunning. I had to share it with you guys. And I've been doing a lot of work with just planning for Get the Dirt and getting a lot of the information out there for you folks. I look forward to seeing many of you out there. We did a site visit training for Allen County so that their working group and their volunteers and employees are able to do really effective site visits in and around Allen County, and a few upcoming events. Alyssa in Allen County also has been working really hard at creating a compost mixer. And it's going to be a really neat opportunity because it's on a commercial size composting with a local company called Dirt Wayne. So you can see what a large composting facility looks like, and it's on a functioning farm. It's called Wood Farms. There'll be lots of great partners out there and lots of talk about soil health. I look forward to being able to see you folks out there if you're interested, and I'll put a link in the chat too for that. And then I've also got a workshop coming up in Madison County, but it's not just for Madison County folks. It's obtaining NRCS funding for small farms and landowners. So it's helping people kind of maneuver some of those systems and understanding some of the practices and programs that are available through EQIP and CRP. So there's going to be just some really great information. So if you know anybody, landowners or small farmers that are interested in obtaining some of those, please send them the link. So here's the information for that. And I look forward to seeing you guys in the future. Thank you.
Thanks, Terry. Now I guess we'll go to the Southwest, my area. So I have been doing a lot of Get the Dirt planning and prepping just like everyone else. We're super excited to see you guys next week and have that come to fruition. It should be a really great conference. I've also been planning for the Pathway to Water Quality next year. We have a small little group. We actually met yesterday at the fairgrounds to talk what we'd like to see for next year in that soil health area and kind of bring that to life and get some more people involved. So that's exciting to be starting now and planning for next year. Also, the USH Advisory Council, like the others, we went to the Full Hand Farm and did a farm visit. So that was fun. Let's Dig in Du Bois County, they held a season extension workshop where Megan and Heidi Potter with Purdue Extension spoke. It was a really great workshop. Happy for them to have something going. So that was awesome. And then Megan also had her high tunnel build at her farm, a 2-day event where we, hands-on got to put together this high tunnel, and now she has something to work with. So super fun. There's pictures there on the right-hand side. The bottom was the first day where we put the high tunnel up in some rain. Everyone still managed to show up and do some work. So that was really great. And then there's the finished result at the top. So the other pictures are from the fairgrounds where we have our worm bin. We still have worms. So that's a good thing in our compost area. So we're beefing that up too. Some upcoming events, October 10th, this week, Owen County is hosting a Putting Your Garden to Bed. The IDEA conference coming up, USH will be at that as well. So you'll see us there. October 20th, there's a vegetable production with wool pellets happening in Monroe County. I've got a flyer for that on the next slide with more information. October 29th, we're going to put your garden to bed at the state fair. So we're going to host a small event and put that garden to bed, throw some cover crops out, and clean that area up. Also, November 7th and 8th in Davies County, there will be a Landwise workshop. Park credits will be available. The first day is going to be focused on row crop farm. And then the second day will be more focused on small scale cover crops and that. So I also have information on the next slide about that. And November 16th, I've talked about this before, Jesse Frost is coming to Vanderburgh County for a farm vitality workshop. It's a lot of ag professionals, I think networking that day. There is a $10 cost, but you do need to go online and register for that. So here's that information. The first one, the vegetable production with wool pellets, it's on a Sunday, October 20th from 1 to 4. You can contact Monroe County SWCD. There is a QR code on there. but you can contact them to register. I think space is limited, so if you're interested, get signed up for that. There's the information for the Landwise workshop in Davies County. CCSI, I think, is going to be involved like the first day, and then USH will be part of the second day. Contact Davies County to register. Contact them directly. And then here's that Jesse Frost flyer, QR code to register. Registration is still open. So there's all that. And now we'll jump to the southeast.
And I'll share for Megan. So there is a lot of overlap here as well
I'm
with some
here.
of the. Oh,
I'm
you're
here. I'm sorry. Yeah, there is a lot of overlap. I enjoyed a couple of our team members coming, and so it was really nice for that high tunnel workshop. Casey joined, and she was one of our fearless workers in the rain. And at the bottom, you can see that crew that we have that was left over on day 2. I think the rain might have scared people off on day 2, but we all had a really great time, and I just really appreciate the community support that came out. Nifty Hoops was amazing. Their crew was just so patient, and I think we all had we had a really great educational and fun time collaborating together to do something that we were all interested in learning about and experiencing. Other things that are happening in the Southwest, we had a great workshop with the Crawford and Orange Soils working group to install 3-bin compost system at the Paoli Community Garden. I attended the Midwest Mechanical Weed Control Day through Purdue at SciPac, which was really great, not only to see so many people out, but to see so many small scale farmers out, and to see that Purdue was focusing on that audience as well. It seemed like about half and half when it came to that mechanical weed management demonstration section of the day. There was lots and lots of small scale tools available. So that was very cool to see. I got out into the field with some site visits, and then engaged with Lawrence Crush working group at a fall gardening workshop at the Bedford Community Garden Park. Presented for the Let's Dig workshop down in Du Bois County, and then like the others, we're at the advisory council at Full Hand Farm. Some upcoming events, obviously Get the Dirt is coming up October 16th. Then October 28th, I'll be doing Putting Your Beds to Bed and Feeding Soil Microbes at the demonstration garden in Jennings County from 10 to 2. That'll be our last at the demo garden for the 2024 season. So I'm really looking forward to that, and hopefully seeing some familiar faces there. October 28th, we're doing cover crop success in the garden at Switzerland County Public Library. There's going to be a seed swap there too. So if you're interested, it starts at 5:30. And then on November 7th, I'm going to also be doing another Putting Your Beds to Bed in Jefferson County at the Soil and Water Conservation District office from 5:30 to 7. And then don't forget, it's grant writing season. So if you're interested, SARE deadlines are fast approaching, as well as, I believe, either today or tomorrow, the Food Animal Concerns Trust micro grants are opening up for 2025. Those are usually micro grants under 3,000, I think, for implementing sustainable practices for animal production, essentially. Those funds can be used for things that many other grants cannot. So, cost of infrastructure, for instance, movable poultry netting, or creating, small buildings, that sort of thing. So be on the lookout. Next slide, please. And I just wanted to share some photos from some of the events I went to. On the left, the top and bottoms photos were from the Mechanical Weed Control Day. There was a great representation of how to utilize a 2-wheel walk-behind tractor on on how to not only prepare ground for cultivation, but also to terminate cover crops, which was really great, and even how to plant using a paper pot planter. In the center at the top, I went to the Purdue forage management field day, and they had this really fantastic tool. So I definitely recommend you clicking on that QR code. It takes you right to their toxic plant library, along with pictures for identification and things to look out for that plant with regard to animal symptoms if consumed in quantity. So I think that's a really great tool, and I plan on having this little card on my table for future events because I just think it's fabulous. Underneath that is a picture of my fancy new high tunnel at night. I just thought it was so pretty and new sitting there in the field. And then on the right at the top is a picture of us at Full Hand Farm. Genesis and Eli are always such generous hosts and do such a fabulous job. They're really some farm heroes in Indiana that small scale diversified farmers should be looking up to with regard to their conservation practices and the fact that they've been in it so long. So way to go for them. And then at the bottom, a shameless selfie there for digging in the dirt at the Bedford Community Garden, where my friends at the Lawrence County office put together a lasagna bed, and we were just digging through it and looking at the soil health that was really evident there just after a season of decomposition with cardboard and compost and leaves and twigs, and it was really, really impressive. So that's all I have. And don't forget, everybody, to check out the website for lots of links for future events, as well as lots and lots of resources to access. Thanks.
Thanks, Megan. We are at time, so if there are any quick partner shares that anybody would like to throw out there, now's the time. And if not, our next meeting is November 12th. I'm not sure what our topic is yet, but we will meet in November. This will be recorded and sent out here shortly, as well as all the resources. So thanks for joining us today, everyone.